Then I went inside, then only I came to know all people they don't know about ABCD, about data integrity. They don't know about anything. They all are scientists. I'm the floor guy, right? These guys are Oxford University guys. I'm a state board guy. So I'm entering into the Oxford University now. The FD asked, how do you do? What is written in the procedure? How do you document?
Correct.
So I was explaining first day, it is going well. Second day, we both are like friends. And FDA got 0483 with 2 microbiologists.
Something I've never heard of.
Pfizer is a really great organization. Only up to Monday to Friday we need to work. And Saturday, Sunday everybody will get off. And you can speak up, the name of speak up culture, you can speak with anybody. In Indian company, you say, I need this time. You have to do it. Everybody is thinking, if you want to grow in a microbiological career, for youngsters, they are thinking, you can grow any level, even you got US government job also. So microbiology has that platform. Current generation, what is the problem I'm seeing? The focus is missing. Why focus is missing? Because of the social media.
Hello all, this is Prashanth here from Pharma Now, and join us for this interesting and exciting podcast with Dr. Suresh Kumar, GM Quality Assurance and also Sterility Assurance from Sun Pharma. Welcome to the podcast, Dr. Suresh. How are you?
I'm fine, sir. Thank you for arranging this discussion with me.
Okay, sir.
So
welcome to the podcast.
It is very, very nice to talk with you.
Yeah. Yes, sir. So today we are, in this event for me. So what do you think? How this entire event is going on, and what is your takeaway from this event today?
If you ask me, by God, as far as my concern, because my passion is microbiology, but exclusively for microbiology event arranged by farming, like me, like microbiologist, it is one of the great, positive tonic. You can say it is a great, motivational factor.
Yeah.
The reason behind that, the people really in a earlier era, 10 years back, if you see, 10, 15 years back, if you see, the microbiologists are just a department in the quality control. They are neglected. They are they have to report to quality control chemists. Then they will reach to the next level.
Yeah.
So it's kind of even if you go to, shall I speak little more? Okay.
Yeah, definitely.
If you see, if you go to the walk-in interview in the beginning time, in the 5-10 years, if you see, means I'm telling to you, 2002 to 2010 in between.
Okay.
If you go to the walk-in interview anywhere, so first they will ask, who are the microbiologists you leave?
Okay.
So who are the chemists you sit here?
Okay.
Who are the quality assurance guy you sit here?
Okay.
But for us, you are making one event exclusively for microbiology. It is really amazing. So attending such, such conference, again, it is making me actually, personally, I need to do little more in microbiology. I have to contribute, I have to learn.
Yeah.
Both the things, actually, it is motivating factor. Really, really motivating factor. This kind of, conference should happen everywhere in India. So it is going to make our Indian microbiologist more competent than, across the globe.
Okay. Thank you, doctor. That's great input. And I also strongly agree with you, like considering even my initial days and the way now at least the platform, what a farming like, event is providing is simply amazing. That gives a lot of opportunity both for learning and also doing the right connection in the industry, for overall growth of the person. Okay. Thank you, doctor. So also moving on, can you please tell us, how you started your education in microbiology and how your entire professional journey has been?
Thank you. Well, and I started, I have completed BSc and MSc in microbiology. I passed out 2002 microbiology. from Tamil Nadu, Manolmuniam Sudarnar University. My college name is Sri Farmakalyani College. That is one of the very well-known college in southern part of Tamil Nadu.
Okay.
So this particular college, as a government-aided college, you have to pay for only 2,000 rupees per year. Even your scholarship, you can complete your master degree actually. But if you want to join over there, minimum you should have distinction, and also you have to, now currently I don't know, but in my time, if you have minimum in BSc 75%, you can able to get a seat over there.
Okay.
So such a wonderful college. So I completed microbiology. Then I thought I have two options. I have to go to professor or I have to go to industry.
Yeah.
So some of the professor, they actually directed me. They told, you go to industry, Suresh, your life will be very good. But I don't know anything about industry and I don't know Hindi also. Just I have microbiology. I came to directly, one of my friend actually, brother staying in Vapi.
Okay.
So just we came by unreserved ticket from Tamil Nadu to, in Vapi.
Okay.
So that is really, amazing actually. But then I came here. So initially I started, because I was searching the job here and there. Then I initially got a job in Cadila Pharmaceuticals as a microbiologist. So that is a bulk manufacturing unit. So there I do water testing and, mainly it is a water testing, API testing actually I need to do. So I was, I'm just learning the pharmaceutical. Then I thought bulk drug means the scope of microbiology is very less. The people, because I'm getting little bit knowledge. Okay, bulk. Then we have formulation also. So then I thought, why I should not move to formulation? Then I tried formulation. I am not getting anywhere.
Okay.
But I got an opportunity in bulk sterile.
Okay.
So it is a bulk non-sterile. Then I got a chance to work in bulk sterile. That is 1 kilometer away from my Cadila Pharma to Wockard Limited.
Okay.
So but I don't know.
For you.
Yeah, I got a chance over there.
Okay.
There actually I started learning the microbiology.
Okay.
Because the sterile, the importance of microbiology is were too high than bulk non-sterile. So there actually during FDA audit, they ask more data from sterile microbiology laboratory. So I thought we have more respect here than bulk sterile. So I was working over there. Then one of the senior most person, he moved from this bulk sterile to another company bulk sterile.
Okay.
Then he made a call to me. So
again, you got one more opportunity.
One more opportunity, that is Orchid Chemicals and Pharmaceutical.
Yeah.
So there I got a chance actually.
So sorry, sir, this was in Chennai?
No, it is in Aurangabad.
Aurangabad. Okay, Orchid in Aurangabad.
Okay. So because in the beginning of the career, I changed very frequently. After that, I stopped actually. I don't know that also. Because he called me, Suresh, can you come here? Then I just went. I resigned here. I joined Orchid Chemicals and Pharma. That is also bulk sterile. Same setup. What I have in Wockard, the same way of, work and everything. I was working over there. Then I thought, I'm working bulk sterile, non-sterile, then bulk sterile, again bulk sterile. Why I should not go to formulation? The same way again, I went to Orchid. Again, I got opportunity to work in Wockard again in formulation in Aurangabad. That is also very near. Companies are near. So I got an opportunity over there. I moved from bulk sterile to formulation.
Okay.
So this particular formulation actually because I got because I am a very strong analyst, basically. I do testing every day. In a 3 organization, I was an analyst.
Okay.
So when I came to this particular organization, Wockard, there I have to do analyst as well as I have to make SOPs, procedures, trends, those things also I need to do.
Okay.
So I was started doing that day by day. I understood whole laboratory in a 4 to 5 years experience.
Okay.
What happened? One great thing happened in my life. So me and my one more colleague is there. Now he's working in Citus Hospital as a microbiology head. We both are working together. He's from Gujarat. So suddenly FDA audit came.
Okay.
Announced audit, but they came here. She is Dr. Barbara Paul, more than 22 years experience in FDA.
Okay.
Very strong microbiologist, PhD also. When she came there, actually, one of my manager was there. He is afraid. to speak in front of her.
I understand, sir. I get it. Yeah, I know that feeling. Yes.
He's afraid and he's escaping from the audit.
Okay.
So management put me in front.
Okay.
That time I was bachelor. I daily used to go gym. I used to have everything. So I don't have any fear.
True.
Okay. But the basic main reason is I was a strong analyst. Then I started I'm making all the procedures here also.
True.
So I know the analysis, I know the procedure. So this is a greatest strength, I don't know. But when they put me in front of auditor, I started explaining to him, auditor very casually. Auditor started believing on me because I'm analyst.
Correct.
I know the floor, each and everything.
True.
Procedure also, I'm revising the procedure. I know the procedure ABCD also. The FDA asked first time, how do you do? What is written in the procedure? How do you document? The 3 circle I know very well.
Yeah.
Because I'm an analyst.
Yeah.
I'm a procedure maker. I made a format also how to make for me easy.
True.
So this circle I already closed. FDA is required that thing only that time.
Correct.
So I was explaining first day. It is going well. Second day we both are like friends. That day I discussed very nicely with him. Whatever I am saying, she is believing.
Okay.
This fellow is telling the truth only. Because whenever they are asking anything, where it is written, yes, this is the SOP. Where did you document? This is my document.
Yes.
And you explained the analysis. I explained A to Z.
Yeah.
So this total loop I closed. That time I don't know. I'm closing the loop. This is the requirement for FD. I don't
Connecting the dots.
So what happened? Then they understood and FDA got 0483 with 2 microbiologists.
Something I've never heard of.
2 microbiologists very strong.
Okay.
Other people are very normal.
Yeah.
Only 5 years experienced person, he is batting with a FDA inspector. This information went to across Vocard Limited global corporate.
Yeah.
And after that, what they did, wherever audit happened, they used to send me across all the sites. So one more interesting is I would like to share if you want to. So I went to Batti. That is the first time USFD audit.
Okay.
There, the microbiology manager, he scolded that microbiologist. He knows in and out of the lab. He left next day. No one is available.
Oh my God.
Only 3 people we are working 3. They put me as a corporate microbiologist like that. They put me in the floor 15 days before.
Okay.
I am understanding laboratory completely. So then after that, audit came. Here also same problem. When auditor enter inside the area, the microbiologist went and sit near to incubator. He is not coming. He is the lead microbiologist. He has to explain.
Similar situation what you face.
It is very bad situation. There I know everything.
Yeah.
I was a procedure making. I am doing everything. Here he only knows everything, but I am in the spotlight now. This is literally different, but I understood the procedure overall. So I started explaining what I know because I read all the SOPs in the 15 days. Based on that, I started explaining. So then when I started explaining, the manager also came to me along with me.
Okay.
Then he came to me because he is explaining. Now little bit situation is controlled. So he also came along with me. But then we both actually, we explained very well. Audit went 0483.
Okay.
The second time, it is the confidence. So these 2 experiment, what I understood actually, how to face FDA inspection. So this information knows by one Vocard manager. He went to Lupin Limited. Then he called me because they have one opening. I sent resume to him. He took my resume and he got good feedback about me. Immediately he called me. So then I went to Mandidip Lupin. So that time I was newly married. I went to there. So we have, my daughter also born that time.
Okay.
6 months, they couldn't able to adjust the climate because entirely different. I am from the south. It is entirely different. Then what I thought, again, we tried to go to south.
Okay.
So that time, one of the big pharmaceutical, that time they are manufacturing, Aurobindo is setting up very big facility in very big sterile injectable facility in Hyderabad.
Okay.
So I just attended the interview through video call. They selected me. Again, I went to Aurobindo. In Aurobindo, What is my experience actually? There only I started working in different department. So I joined in the laboratory. It is a new facility, completely lab setup. From starting to first USFDA, first Anvisa, I worked over there. Then I went and I told my site quality head, I am saturated. You give me some other work. So that kind of attitude started that time. Then I asked him. Then
Very rare, approach, even in a working environment. Very few people go and ask, give me extra work.
Yeah.
That is something incredible. That's a great trait what you have.
I asked him, you give me work in the quality assurance. I have done in microbiology. Because nothing. Still learning is there, but that time mindset is like that. Because I started from the scratch, and there every single equipment I involved, every single procedure I involved. Then I went to this first FDA, first Anvisa, this approvals, all approvals we got. Then I went and I asked, you give me in quality assurance. Then he told, tomorrow onwards you come in quality assurance. But I don't know that time also. They are giving again microbiology to me, that I know. They told me, you go to IPQA, in-process quality assurance, you take care of IPQA, as well as you have to do environmental monitoring in the area using the M-Form guys.
Oh,
it is a big task for me.
Yeah.
IPQA guy, because Aurobindo took a decision, IPQA will do the floor environmental monitor. IPQA, most of the people are M-Form guys. I am the only microbiologist. Then we put 3-4 microbiologists also from laboratory. Then I started teaching M-Form guys how to expose the plate.
Okay.
I started learning from the in-process checks, BMR review, PPR review. Those things I started learning.
Okay.
So one side I'm giving training to them, one side I'm learning this also. This is a good experience actually. So around 4 years I were there. I was, I worked over there around 4 years. In the 4 years, so again one managers, he left from Aurobindo to Pfizer, Aspira that time. It was Aspira. He went to Chennai. He just made a call to me, Suresh, we have one walk-in interview in Hyderabad, Begumpet. You can attend, some of the positions are open. Just morning shift, I just changed my shift to second shift. I went to Begumpet interview. I attended, I came to company again, second shift. So I thought just like that, I went and I have given my interview. No preparation, nothing. There actually, sir, what I got actually, they selected me actually. So here I am working as an assistant manager because I joined in Aurobindo as a senior executive. Then one time I got a promotion. Then I resigned Sun Aurobindo, but the Aurobindo management told, we are not going to leave you. Whatever you want to do, you do it. You be here.
Okay.
You don't have any thankful career. You don't have anything because you learn many things. Because I worked in laboratory, then I worked in quality assurance. So they have given platform for me for learning for platform and promotion also they were given. And they told, we cannot leave you. Not possible. One great thing happened in Aurobindo. What happened? This is my personal growth actually. And some other biofilling bacteria, they could not able to control in the water system.
Okay.
So I did a lot of work actually in that.
Okay.
There finally we controlled. We identified all the issues resolution. So then Cited came to me, and Cited means overall Cited. He's a very great guy. So he came to me and he told me, Suresh, here you put a lot of effort. Hours together work I put to establishing the water system, to establish the good water system. So he told, you got a you may get promotion, but we cannot do more than that. What you do, why can't you go for PhD for this topic? So that time, I just thought, whatever data I did here, I took all the data. I made a synopsis. I just went and I approached my university. I would like to do PhD for this because I did everything here.
Okay.
Formally, I have to make in outside everything. I have full data with me in the back end. So I just went and I just, there we have the very, very, very fair interview for outside student.
Okay.
So they have a big panel. They discuss with me. Finally, they understood. I know clear problem. This is my problem statement. This is my cause. This is the thing I did. You are going to get solution here.
Yeah.
It's very clear communication. They selected me for part-time PhD.
Okay.
So their PhD started. So PhD, then I thought I cannot do with this company. I got NOC from my organization. They have given NOC to me. So directly I went and I one more laboratory in Hyderabad.
Okay.
I just started working over there in my weekends, in my evening time.
Okay.
I started working, little fine tuning of my project. Then I submitted the thesis over there. That time, that is one of the commendable theses in my university.
Okay.
They have given the rating. The thesis rating will be there.
Yeah.
One rating for us is commendable. It is very rare to they will give.
Okay.
Second thing, my professor applied for the Indian patent.
Oh, okay.
So it went for Indian patent also application. Okay. So this is the way I got a PhD within 6 years because I struggled a lot. 6 years I got a PhD. So that is one side. This is a Aurobindo. Then I went to Pfizer. Pfizer, really amazing experience in my life. Pfizer, I joined as a deputy manager because here I was working as a assistant manager. There I joined as a deputy manager. I joined in a complete in-process quality assurance.
Okay.
So now there is no microbiology here. Cut off here. Microbiology in-process quality assurance. So there when I went inside, a lot of investigations were pending.
Okay.
Why the root cause is not identified, problem is there, production is not supporting, micro is not supporting, IPQA is not hearing the words like that. Then I thought, first we have to unite these guys. Okay, we have to do the 3C. Connect, collaborate, and one more thing is there. So we used to say, communicate, connect, communicate, collaborate.
Okay.
So first I thought connect to all the people because everything is happening in silos.
Okay.
40 batches are pending to release. Investigations are open. QA is saying, my team is saying, they are not giving any root cause, they are not giving any proper document, I cannot release. I am the superior. Production is saying, I explained multiple times, they are not doing. Then I put everybody in one room. You sit here, Pele. You tell the problem. Okay. Then you tell your expectation. Quality assurance, because their quality assurance is very power because they always involve in the process in Chennai.
Yeah.
That's the greatest thing. Other things they used to monitor. Here, these guys are involved in the process.
Okay.
My team member, they know the solution also. But only the problem with, I cannot say directly ego. It is the superiority. Means I am superpower like that.
Yeah.
Then I asked the team only, you tell me what we need to do. My team told, this is the way we do it. Then I asked production, why can't you do this one? So this problem. Yeah, you think in a different way. Then both of them, what is the easiest way? What is required? Not easy. It is not a comfort zone. I'm saying what is required. We took a decision, 40 investigation within a month. All 40 investigations are approved. The same investigation reviewed by MHR auditor. There is no concern also.
Oh.
So management thought, what is happening over there? So the particular block where I was leading the quality assurance team, the block always has bad name. They will not work like that. Both the team. But they are the highest money-making block. So that is in a pressure is more.
Okay.
They understood within 9 months over, everything is going smooth. Then they started 2 programs. One is manufacturing quality assurance program. Another one is investigation certification program.
Okay.
It is a very genuine program from external party, Quintels. They came and they monitor and they give the program. One is 1 year program. Another one is 6-month program.
Okay.
They have multiple classes. We need to complete the quiz, very genuine way. If you pass, they will pass. Otherwise, they will say, this fellow is not qualified for manufacturing quality assurance.
Okay.
So the way it is going on. So I qualified manufacturing assurance also. I was qualified for investigation program within 4.5 months, 9 months program, 4.5 months I qualified, both.
Okay.
So management was completely impressed. 11-month career, 11 months is completed in Aspirations. They promoted me from deputy manager to manager. And they created a separate department for investigation team. You are the investigation team for complete site. You will give 7 senior most people. You handle across all the, from laboratory, manufacturing, engineering, warehouse. All investigation come to my department. They review. We have each experts everywhere. I was leading the department. I am reporting directly to my manufacturing site quality head.
Okay.
He is from foreigner. So I was reporting there. So over a 1 year, 8 months, I was working like that. In the meantime, in Pfizer, they want to set up a global microbiology method validation laboratory.
Okay.
They are charging for 6 months. They are charging for many candidates. So directly I applied one day. Someone came to me, Suresh, why can't you apply here internal job? I just applied. They called me interview. That is also a very big panel. I had an excellent communication that time, and I had PhD that is working that time.
Okay.
So they need a guy PhD. Then I told, I am in a quality assurance. If I want to come back again microbiology, you give me minimum DGM. Just like that I asked. Means associate director.
Yeah.
I was working as a manager. I asked, if you give me associate director, I will come. Otherwise, I'm not coming. Like that. Because I want to deny. I would like to continue here. That one guy is impressed anything else that I don't know. He told, finally, we're ready to give. It's amazing in life turning point.
Okay.
And HR is actually against to this accomplishment because other colleagues will think very badly.
Correct.
Because someone is promoting from manager to associate director. This is entirely different scenario. It is very tough. We cannot give. They told, we need a person who knows the manufacturing, who knows the laboratory. This is the guy has the knowledge from the floor as well as in the laboratory.
Correct.
We need him. They have given clear justification. And that is a different, within the Pfizer, different entity.
Okay.
So, we are not going to disturb your manufacturing sector. It is an R&D sector. We will take him in R&D. You don't worry about it. In R&D, in PhD, whatever we are going to pay for him, that is a very lowest salary.
Okay.
It's not a pay or something lost to organization. But he's asking associate director, we need to give it. So they have given the associate director. That's a miracle. God blessing. Something we can say it is a miracle. So I got an opportunity. Then I went there. Then I went inside. Then only I came to know all people they don't know about data integrity. They don't know about anything. They all are scientists.
Understood. So
They are
it's
all
a pure R&D setup.
Pure R&D setup. We need to do method validation for the GMP site. So I need to make them now a reality because 1 or 2 colleagues, if they drink tea, they will keep the cup here up to evening. They will not take out also. Such people, they were given to.
Very highly non-functional team we have to work with. Correct.
Drink the tea, they will keep here in the
Understood.
evening. They can able to take it, they can put into test with this. This is the word.
Understood. I got the message.
I understood very clearly because I basically, what is my motive factor if you ask me? Go and do the work in the floor.
Yeah.
What is required there, you will come to know. You
Yeah.
can understand well. I'm the floor guy, right? These guys are Oxford University guys. I'm a state board guy. So I'm entering into the Oxford University now. You understand?
Understood.
It's a big challenge for
Yes.
me in a career point of view. So then I thought where I need to work because first I have to make the people accountable. I have to make them understandable. But after that only I understood the team is ready to work. They need a direction.
Okay.
The direction is missing from the earlier management.
Okay.
Then I started, what I did actually, every day I used to put in a, visual what we can say, whiteboard. I used to write everybody what is the work pending, what is the plan, when we can do. It is a 15 minutes meeting. We started with every person, I make them occupied. I started taking the work.
Okay.
Whatever pending you have last 2 years, bring everything. One by one, because what is the problem is there only one simple problem. They want to make any decision, they will go and ask the global guy. And the global guy will be available once in a week, 40 minutes. What you can understand, I told I will take a decision. I am the department owner. I will take the decision, whatever will come, it will come. Like that I took and I aligned with the floor QA. I aligned with the management in the site.
Okay.
Then I started putting CRUSC. What is my decision, what I'm going to do. That I made very clear, clear things. This week, what I'm going to do, what is my decision, what is the thing that I communicated through mail to my boss, and we will discuss on Friday. If you have any concern, in between you talk with me. So, the communication to the top and communication to the bottom, I clear first. Then cross-function also I aligned. So after that, we started working very well. We cleared many projects. Then they told, you renovate the new microbiology laboratory. Just you make it complete new laboratory. They have given the fund to us. So that time the qualification means R&D means I do only testing. I do only this. I told all qualification activity. My team will. I'll teach my team. There is no other manpower is required. Give me. Like that I told. Man, how you can do? You give me, boss. Whatever equipment we qualify.
True.
We started qualifying all the microbiology laboratory. We qualified complete laboratory with my own team. There is no external support, nothing. We qualified ourselves. So that's really amazing point for management. They appreciate a lot. We have now 2 new microbiology R&D laboratory. It serves for Australian site and also US2 sites.
Okay.
So that's, and Indian sites also. If anything related to method validation queries or regulatory queries, we used to involve. So that level of contribution we started.
Okay.
So in the meantime, Vizag, the head microbiology position, the team size is around 110 members.
Okay.
One of the site head moved from Chennai to Vizag. So he made a call to me. He just asked me, Suresh, if you're interested, you can come and take this opportunity.
Okay.
So then I moved to Pfizer, Vizag.
Okay.
So that I promoted as a GM. So this particular journey from Pfizer, I joined as a deputy manager within 7.5 years, 7 years. 6.5 years, you can say, I promoted as a GM. So that's a really great journey. And in this journey, if you see different stream I worked, quality assurance, investigation, R&D microbiology, and site quality manufacturing microbiology setup. So here again problem is different. People are 110, data integrity problem, technical issues, and there is no commitment at all. Pfizer is a really great organization. Only up to Monday to Friday we need to work, and Saturday, Sunday everybody will get off. And you can speak up the name of speak up culture. You can speak with anybody.
Okay.
There is no fear inside. I have to complete the work. I can give the reason, real reasons.
Okay.
So this setup, I need to work sophisticated way. I need to get more outcome also. Then I thought here, if in Indian company, you say, I need this time, you have to do it.
True.
But here it will not work. So what I thought, go for simplification, go for what is not required.
Okay.
And you just take all the waste from the system. Automatically, you can get the good system.
Correct.
So that is one of the greatest agenda. The second agenda, what I did actually, data integrity I removed completely. How I removed this is also mainly technical only. Many people are thinking administratively you can make it data integrity, but rather than you go with the procedure in a very simplified way, keep the people with higher level clarity, you can reduce 80% of data integrity.
Yeah.
So that I did. That experiment I did. So after that, one biggest problem with the Isolator. The Isolator, we got a continuous EM excursions.
Oh, okay.
So that solution brought by me and our engineering team, that's very interesting. Actually, if I go any interview, I used to say that story only first. Everybody will be impressed. So that was happened there. The arresting the problem of Isolator by providing the solution from in-house engineering. So we got a solution from our side and we arrested.
Okay.
There I was working there. Then I thought I am working long time in Pfizer. Why I should not see some outside opportunities? That may be a wrong decision, but okay. Then I thought, already they have given in between, inside only they have given all the opportunity. I don't want to see outside actually. But the destiny, the time, it always has some, a path forward. So then Aurobindo Pharmaceutical, because earlier I worked in Aurobindo, they asked me, we have the position for 13 formulation sites as a technical microbiology lead. So I got an opportunity over there. I worked there around 1 year, 8 months. Every month we used to have USFDA audit. Every month we have many things. So travels are more. Then I thought the corporate sitting in, writing in the policies, giving the technical it's not completely matching with my. I'm a floor guy. I need a team. I need a people. So in corporate microbiology, I have only 3 members, and 3 members also we have to travel across all the sites. Any problem, any audit, we have to go there. That is also one of the good experience. That is also experience. But corporate, I thought this is not my cup of tea. Then I switch over to again Sun Pharma. Sun Pharma, they have very exclusive role.
Okay.
It is not like just a microbiology role.
Mhm.
They told me, you are going to lead us floor microbiology,
Okay.
process microbiology. It is completely related to process microbiology, Suresh. You are not going to take care of laboratory at all, testing, yeasting and all. You are not going to take care. You have to go with the floor and you have to do it. That time, one interesting thing, I met one scientist in Bangalore airport.
Okay.
We both had a very good friendship, because he is a great scientist. He is working long time in University of Tennessee
Okay.
in US.
Mm-hmm.
He needs one experienced microbiologist. So then he arranged the meeting. He arranged interview through US University of Tennessee. They selected me.
Okay.
So I have 2 offers. One is from Sun Pharma. Another one is from US with H1B visa.
Okay.
Visa also they arranged.
Mm.
Visa I got. One in my hand, visa is there. H1B is there. That is R&D, completely pure R&D microbiology. Here I have manufacturing these things. So it's a family decision. Why I'm telling this, I got a US offer like that, because anybody is thinking, if you want to grow in a microbiological career, for youngsters, they are thinking, you can grow any level, even you got US government job also.
Yeah.
So microbiology has that platform.
Yeah, true.
So that's what I'm just explaining. Otherwise, I can skip that part. Okay. So I came to here. Then I thought family told, we are children are teenage now.
Mm.
And now if you go there and why can't you, Suresh, would you please rethink? So then I thought we go to, again, the same, Sun Pharma. The same field, same.
So that's a floor microbiology.
Floor microbiology. This is a process microbiology.
Understood.
Here I do, my team does every day, we do actually viable monitoring, non-viable monitoring,
Okay.
media fill visual inspection,
Mm-hmm.
complete media fill program.
Okay.
It is being designed or across all the, sterile, our manufacturing unit, our team actually,
all the injectable sites.
All injectable sites.
Mm.
So our team is doing. Really, you can see everyday problem. You can see everyday solution. This is entirely different and a lot of importance for microbiology in process. So that learnings actually I contributed also based on my experience. So currently, last 3 years, I'm working in Sun Pharma. So we faced multiple audits, except USFDA, all audits we cleared. Mm-hmm. Even we implemented completely this across all the blocks, 6 blocks we have, and now currently 5 blocks, all 5 blocks, contamination control strategy. What is written in the UNX one? The same thing actually we implemented across the site.
Okay.
So contamination control strategy is also part of my team actually.
Okay. CCS.
Media fill program, contamination control strategy, floor microbiology, floor viable, non-viable monitoring, and any investigations which is related to sterility assurance. So that actually our team is to take care.
Okay.
Second thing, sterility, we established sterility assurance. When I came here, first sterility assurance department, we established in Sun.
Okay.
Sterility assurance. Every month we used to have the discussion with very high level people related to only sterility assurance.
Okay.
It is very new to the, not new, but it is in the Pfizer. But if you see across Indian Pharmaceutical, exclusive sterility assurance discussion has many, very, very few companies.
Okay. Sir, I'll just, just wanted to understand. I think many of our viewers also will be interested to understand this. What is the difference between your QC microbiology and sterility assurance?
Yeah, I will come to you. Because in the sterility assurance is a big things.
Okay.
It needs everywhere. In laboratory also needs and floor also need. Mm-hmm. But if you see sterility assurance is required more in the process.
Okay.
Rather than testing.
Okay.
Testing only you need to control whether I'm going to make mistake. I should not make any mistake. Because of me, there is no contamination should be introduced in the testing.
So you
that
should get the results of the product.
That can be done within the block, within the particular zone. If you go to sterility testing session area, you can control.
Yeah.
But in the process microbiology starts from your material entry tool, your dispensing, your tunnel sterilization, then your filling process, your intervention, your container closure. Then it goes for, visual inspection.
Correct.
It is the end-to-end process in a sterile. You need a microbiological conscious mind or assessment is required. If you ask me, two people are very, very important in sterile aspects. One is microbiology knowledge. Second one is engineering solution. These two people, they have to work together for each and every process. So process microbiology is the primary microbiology now currently, because earlier, if you get one contamination in sterility testing, you need to think over your batch, whether you need to keep or reject. Today, if you get one bacteria in the filling zone in the sense infit and table, filling zone and stoppering station, or if it is a lyo, half stoppered vial goes up to loading.
Yeah.
Till the way you get one bacteria in one settle plate or one air sampling or one surface monitoring, we are rejecting the batch.
Oh my God. Okay.
So, what is the importance of process microbiology today? Why FDA is going behind with the process microbiology today? The reason is they want to control in the process, not the laboratory. So, the sterility assurance more focus on the water utility monitoring, compressed air monitoring, environmental monitoring, NVPC monitoring, and also aseptic behavior and aseptic techniques.
Okay.
The techniques comes under intervention.
True.
Aseptic behavior controls personal behaviors.
Yeah.
So the human is the big contaminant, because you know that 1 square centimeter you have millions of bacteria.
Yes.
He is the biggest inoculum. He is going inside the area.
Yeah.
Rather than testing, I'm not going to inoculate anything, because now we have the closed sterility system.
Yeah.
Just if you put, or you are going to suck the solution, you are filter it, you are going to see only 20 vials. But here, the big human being, through his technique, through his aseptic behavior, he can contaminate in the big area.
Correct.
In the filling zone.
Yeah.
So that's the reason sterility assurance plays very key role in the process. Example, I'm telling you. One person, I'm coming into the filling zone. I'm going for gowning today. I have to go to the process. First, I remove my street clothes. There itself, you wash your leg properly. You sanitize your leg properly. Then you go with the primary gowning. Then you go with the secondary gowning. Then go inside. Even though if the person mistakenly here, if I'm working in agricultural land, I'm coming directly, I'm not washing my leg, I'm doing my gowning. If I go inside the grade B area, you can get the recovery of bacillus.
Yeah.
How the bacillus is introduced from the outside to inside area. So through person. Next one is material. I'm sanitizing my material. Some of the material I'm putting into the pass box, then I'm transferring inside the area. If I miss some, sanitization is a manual process, right? If I did not establish properly here, I'm going to introduce bacteria in my grade B area. When you introduce in a grade B area, automatically it goes to grade A area, because we have ORAP system. We don't have any isolator system. For example, ORAP system, conventional ORAPs, the control, the process microbiology mind and knowledge, and every excursion, how you are going to address through engineering solution, that is the game in the sterility assurance.
Okay. So, so if I understand this, the entire explanation, so the pure microbiology still stays within the lab, and wherein you don't want to contaminate the entire testing procedure. You just want to test the product and give the result.
Yes, sir.
Whereas a sterility assurance is not just the testing; it's leaving the testing rest of the things.
Yes.
Correct. End to end, starting from your material entry to packaging. So you include all the utilities, clean rooms, even the people. So entire thing contribute. So controlling this entire thing and ensuring the product safety is sterility assurance.
Absolutely.
Okay.
Absolutely. End to end.
Got it.
But in earlier days, why I am emphasizing here, earlier days, the people, I would say very clearly, openly, environmental monitoring, if anybody is not working properly in the laboratory, we used to put in environmental monitoring.
Yeah.
Why we used to put? Why we put actually? Because there the limit is 5, 3, 5. Put 3, 1, 2. It should be in a trend.
Yeah.
We will ask them. This is the reality I'm telling you.
Yeah.
Earlier days,
Yeah.
you need to make a you need to maintain the trend.
Yes.
You need to maintain the data.
Yeah.
But now the pharmaceutical after 2010, completely the data integrity culture started. Now completely they are focusing on every single colony recovered in that grade A area, grade B area, and also surrounding now. Even they're working on surrounding also.
Mm-hmm.
Literally, you can see now in pharmaceutical in the outer corridor, CNC area, controlled non-classified area.
Yes.
They are cleaning thrice in the injectable facility.
Okay.
Never happen. Earlier only they focus on grade A and grade B.
Yeah.
Other area we will see like that. Now the mentality is completely shifted because every single count is a question mark about your
batch.
Manufacturing, direct business impact, direct compliance issue, direct issue now. So that's the reason the importance is going up now for microbiological things. But only thing what we now pharma, pharma now actually they explain more about automation in future.
Yeah.
So how the automation, because gloveless isolator is going to come. So such things if you come, definitely all the problems will be nullified.
Okay. On that take, sir, like what do you feel like take next down the line 5 years or 10 years? Do you see a scenario wherein the people will be totally out of the complete manufacturing process as such, and it will shift totally towards robotics or something even more advanced?
Yeah, really, what I'm thinking now, completely what you are saying, the robotic, the movement, that the first step it will come, because now we are handling that's my understanding.
Okay.
Because now we are the people, because first we have the open just to door only.
Yes.
So now most of the company they converted into open ORAPs.
Yes.
With wraps actually.
True.
We reached that level. Even existing system also. Then one level up, current all pharmaceutical, they are putting only
isolators, yes.
So now the next scenario, the isolator, the gloveless isolator will come.
Yeah.
So automatically when gloveless isolator will come, automatically reduction, but it cannot happen. All the gloves you remove immediately, 1 or 2 gloves they keep, because it is an evolution, right? One by one will come.
Yeah.
So what you are saying, another 5 to 10 years, after 10 years, if you see, all persons will be out.
Yeah.
But the concept, whatever concept we have, the basic concept, that will be converted to AI automation or something, it will be converted. That is required, really I'm saying, because my experience last 22 years, the person you can teach, but you can see the same mistake with a different person.
Yeah, I get it.
It is really what I'm telling now. One example I'm telling, you open the door, whenever in the grade B, if exposed, you have to sanitize and you have to close it. It's a very basic thing. But the same thing, if you see person to person, they will open the door, then they will not sanitize, they will close. The requirement is you don't open the door.
Yeah.
Starting to end. You cannot open the door. The problem will be arrested.
Yeah.
That's the only thing can give 100% solution. But however, I have seen my experience, BA failures in isolators. Environmental monitoring excursion in isolator also there. So the concepts of microbiology will be remain same, but only the technology will come. The technology will come also. The understanding of the microbiological concepts is always required.
Yeah.
So no worry about microbiological job will be reduced, but we will have different kind of job. But the understanding of microbiology again required for human being. Human intervention is required.
Yeah.
But that is not direct intervention with the product. That is required only technology side.
Yeah.
Like how I have to operate the AI, and what best isolator validation cycle I can able to develop. Because automatically robot cannot develop much. We have to think and assess, okay, this is a requirement, then you feed the data. So I think so that we need to do. But
okay.
In future, once the human intervention is removed and AI is coming, robot is coming, the problem is 100% resolved.
Okay.
What kind of FDA citation, what kind of issues we are facing now? that will be removed. Because even I am seeing some of the auditors are coming in isolator facility currently. My experience I'm sharing with you. Isolator? Yeah. Validated with the BA? Okay, good. Then they are not asking anything about environmental monitoring data. They don't have any concern about sterility. So they are going with a different thing. Your BA validations, your load pattern, those things are okay. If they are satisfied, no problem at all. Like non-sterile, it will be approved. I've seen, oh, facility is good. Okay, which company? Okay, this is a company. What is your gradation? What is your surrounding grade? Okay, it is okay, C grade. So what Annex 1 is saying? Okay, Annex 1 is saying grade A, background is grade C. Yes, okay. Okay, do you have exposure and we have everything? Done. But if you come to now conventional grade A by B, grade A, background B, auditors are sitting more than 11 hours or 12 hours.
True.
Just they are watching like, reptile. Like, reptile means like they are watching 10 years. The person lift the hand, or the person any shadowing or anything. Right. Carbon, we have small dot. What is that, dot or ton?
Yeah.
Take out outside, we have to open and we have to show to them. This is only dot here. Someone kept pen or something.
Yes.
So I'm telling to you, these kind of challenges will be reduced. But the main crux of the information, what I understood being a microbiologist, human is the big contaminant.
Yeah.
When you introduce him inside, knowingly or unknowingly, because so many control. First to control, the glove. I used to give training to the people, my operator. 3G you have to follow. The 1G is glove. Second one is gown. Third one is goggle.
Okay.
If you maintain 3 properly inside the area, you are going to prevent the contamination.
Yes.
So this kind of 3G concept. And one more thing, touch and transfer. I have seen many, some of the media for failure I have seen I noticed actually. I got a contamination from the room push plate. Same contamination, same bacteria recovered from the floor, same bacteria recovered in the your
failure
vial also. So this touch and transfer, how it is going from your room push plate, a door push plate, and it comes to floor, and again it goes to your vial. So that, because bacteria is invisible, but you can only correlate the data, identification data. So here it is moving like that. So if you put a control here, where is the problem, how it is going to B grade, B to A, there if you focus on that, automatically it will be best. Like that, many things in change rooms also. Throughout my career, I have seen many times in change rooms, we got multiple time excursions. So continuously we are exposing the plate. Every day we are getting exposure. We are surprising what is happening here. Nothing happened. What happened? Finally, we identified one small hole happened in the, your panel. The air is going into the panel. It is coming out from your door frame.
Okay.
Lightly it is coming. So inside the air came, the same air returning to the room.
Okay.
This was identified after 1 month. We arrested that problem. Next day, there is no excursions.
Okay. It's a tiny dot, but there's a lot of contamination, a lot of business impact, a lot of commercial impact eventually. Okay.
So like that, many things are there.
So, okay. So, doctor, also you have almost, 22 years of experience in this entire microbiology, career, and also sterility assurance. So, how has been your experience with the regulatory, and starting with your initial days of career, and when you see today, even how the regulatory has changed?
Regulatory, this is very, very, very nice question actually. In regulatory, I would like to say when 2002 to 2010, we can have such time actually in a time.
Yeah.
So it is an announced audit. They will say 1-2 months before, people will be ready in India. Whatever corrections in my document from the last inspection to this inspections, everything we correct. Fact I'm speaking. Everything we correct, or we need change control, we need last time decommitment, what we did, everything we will have answer within that period.
Okay.
So 2010, so one great change in microbiology is one of the company, I don't want to say the company, that company They verified the incubator. How many plates are inside the incubator and what is available in the result, both are not matching.
Okay.
The auditor is going out just like that. Audit is going to complete in the last day. Microbiology manager is literally enthusiastic. He asked the auditor, why can't you see my incubator like that? Confidently. So he thought he will not check.
Yeah.
He came and he verified the incubator completely. Then they came to know both are mismatched.
Okay.
And 10 days before, plates also were placing, were lying on the incubators.
Oh my God.
And fungal contaminated plate also lying on the. So then the day the day the microbiologist change day, you can say.
Okay.
FDA has given 483 anything else in environmental monitoring, and they identified environmental monitoring section is one of the weakest section we can get all the time point.
Every time, in fact.
Before that, there is nothing. There is no focus on environmental monitoring. This is a regulatory point I'm telling you. So that time, the maximum position in microbiology is assistant manager.
Okay.
When this observation came, after that, the position created for senior manager, the position created for DGM.
Okay.
So the importance of microbiology changed from 2010 to 2014.
Okay.
That after 2014, wherever people are going in company, they started giving on the observation in laboratory as well as process microbiology.
Okay.
Recently, process microbiology. The 2014 to 2020 up to, if you see, they more focus on the plate reading. Plate reading means data integrity. Up to 2018 or 19, I can say. They go to the laboratory, first they read the plate. Plate read, one count I put here, but if I see the discarded plate, I may get 2 or 3.
Yeah.
That data integrity they started. And they started watching. Earlier, I'm telling to you 2010 up to 10, they come to laboratory, 2 tests they see the demonstration. One is BET, another one is sterility.
Yeah.
So both are okay. Laboratory round is over.
Yeah.
Then microbiology round, then they go to the trend, water trend and EM trend. They see graphical presentation, everything is okay, done. But now not like that. Now, after that, what they did actually, every simple microbiology sampling, testing, everything they have seen real-time sampling.
Okay.
The same way they shifted 2019 onwards, they shifted to manufacturing.
Okay.
Now the manufacturing, boss, your media fill video you give me your smoke study video, you give me your, then first they will watch currently what is happening here. Give me the actual BMR to me. I have BMR, how many times I took for completion of my manufacturing.
Yeah, all the intervention.
Media fill, how much time I took for my assembling and setup and everything. And smoke study, how you perform the smoke study in the intervention. All three, they are correlating. We have always mismatch now.
Okay. Because each one, I believe that we see it very differently, not in a practical way.
Yes.
But when auditors brings it to reality and sees it in a very real-time scenario, that is when, the initial point what you told, connecting the dots, that connection of the dots will not happen.
Yes. End-to-end process.
Okay.
If you know, as a manager knows, we can handle the audit earlier.
Yeah.
Or up to assistant manager, you can handle the audit. Now, end-to-end process should be known by your operator. And what your operator is doing every day, that you should monitor.
Yeah.
And in front of audit, you just be ideal. They watch everything. If they performed well in the floor, harmonizing the practice within your team, microbiologist or production operator or anybody, if you harmonize the practices and procedure, align with everybody, what they are doing, what is required we need to do. If they are doing, if you can able to demonstrate in front of FD, you can able to demonstrate.
Okay.
It is very clear-cut, real-time monitoring. They are believing now. They are not believing your data, your SOBs. They are believing actually your procedure, your lot of validation package. Those things they are keeping aside. Just they are asking what is your filling today, your filling operation today. They are coming and they are sitting there for filling start to end, assembling to end. The same process, everything they are matching. Wherever they are getting discrepancy, they are monitoring. Then they are bringing your smoke study, bring your media fill. Then they started correlating this. Then they are correlating with the investigation also.
Okay.
Okay, these many excursions you got. Very good. Because earlier, now current scenario, I will tell you. Earlier, if you are not raising the investigation, they started fighting us. Now, you raise the investigation. Okay. Very good. Very good. You raise the investigation. Where is the kappa?
Yeah.
Where is the corrective actions? How do you control?
Yeah.
You cannot go here also. You cannot go here also.
Yeah.
If you raise more investigation, you can say, I am so transparent. Okay, you are very good, transparent. You are not going to say anything. Where is your action?
Yeah.
Where is your patient safety? So, an anomaly monitoring excursions, identifying the root cause without camera, without some evidence, microbiology is very tough to find the evidence.
True.
So they know that gap. Just they are putting pressure on us. One side they are telling escalate everything. Other side they are saying if you escalated everything, give me the solution.
Yeah.
It is a very fair game only. I'm not saying anything it is wrong, but that maturity we need to achieve now.
Yeah.
In especially sterile plant, what I'm doing, whether I'm doing right or wrong, I have to verify properly and I have to keep all the resources. Why in microbiology, another microbiology biggest change is earlier they used to keep with less manpower. They do everything because it is a manual documentation.
Yeah, true.
Automation is very high. But currently, if you want to success in the pharmaceutical, especially injectable, you need to have resources. Resources means your equipment, your people, competent people, everything you need to keep.
Yeah.
If you keep only, you can able to success.
Yes.
The current scenario of a regulatory data integrity with proper trained manpower, if you have in the floor, you can able to face the audit. Otherwise, you may be a very big organization, you may be very big, you have everything, procedure, policies, so many things. There, if you fail, everything will fail.
I also strongly agree with that, doctor, and I also believe it has to do with the company culture also. That will take, not just months, years together, wherein people have to drive it and, make it a reality. That is going to change the, entire industry. Doctor, thanks a lot. You gave a lot of inputs about your entire journey, the work, the solutioning of the problems what you have faced. And, I myself learned so many things. Probably one of the most important thing was connecting the dots earlier of your career, what you were mentioning, how a FDA auditor was asking and how you connected the analysis, documentation, and usually explaining this to the auditor and convincing them. So that was one of the thing. Then next thing is communicating with the people and also bringing the people together. One of the team members you were mentioning, right? So you could solve, so many problems because of that. And also now leading such big teams, I'm sure, you're making huge impact for the industry. What myself and probably our viewers also want to understand is, what is one message you want to give away to us, which, we should take it very seriously and we should work on, because one of the most important thing what you mentioned is, earlier days there was not many chances for a microbiologist, but today when you gave the instance of your University of Tennessee, and the job offer from Sun Pharma when you were mentioning, so that is when you mentioned today's day microbiologist can go to any level.
Yes.
There is very high probability, right? So what is the takeaway you want to give to our viewers, so that that is something they should seriously look into and work on that?
What I can say, because focus. I can say only main thing is focus. If you focus on your work, if you committed your work and your punctuality means attendance. You focus on attendance, you committed to your work and focus. So the current generation, what is the problem I'm seeing? The focus is missing. Why focus is missing? Because of the social media, because of, the reading habit is very less now.
Yeah.
Earlier days, what we do actually, sir, if I see one problem in a laboratory or anybody asked any question, I go and refer all the guidelines.
Yes.
Where it is written, whether I am doing right thing, the law, SOB is law.
Yeah.
The law I made from which guideline, where it is written. Now you put Google and you just verify and do it. No one is reading now very depth way. So my only thing, you focus on your work. You have very less knowledge today, but when you focus on your work day by day, focus on the floor, focus on your own work. Don't be distracted when you enter into the enter into the work area to end.
Yeah.
Be with work.
Yeah.
Automatically, everybody will get knowledge. Everybody will get appreciation. And microbiology is booming of anything else now. So you can enjoy your career because what we studied, we are going to apply now.
Yeah.
That is one of the great boon from God. Other people are not like that. So we have that platform. Only thing is focus. My only advice to the people, not advice, my own way of growth is focus.
Yeah.
So now this Pharma Now event actually is making me more focused.
Okay.
Because really I'm telling you, because we have one exclusive conference for only microbiology.
Right.
This is actually, amazing.
That is something incredible, right? Me being a microbiologist, I always get excited. A platform which is provided like this.
Discussing microbiology concepts inside.
Yeah.
It is, really amazing. So what I can say, when you do any work, you understand first. Just to doing work, anybody can do.
Yeah.
With a microbiological conscious mind, you just approach the issues. You will enjoy the learning, and you can contribute more also in organization, and you contribute for your own, for your family also. My main, things I can say, current generation in India, they have to focus more.
Yeah.
The focus, the distraction from the social media, distraction from anybody words, distraction from anything, you leave it. Microbiology gives everything to you.
Yeah. It's the focus, and probably I'll add one more, the patience. We need to have both.
Yes.
Then I believe, as you rightly mentioned, and all the opportunities, you yourself are a great example.
It is a big ladder for everybody.
True.
Big ladder for anybody actually. From the, I completed MSc sufficient. You come to industry and you focus on day by day, automatically everything will happen for your own accomplishment. Second thing I can say, it is a noble job when we are doing the industry. When we are in pharmaceutical industry, specific to injection, it is directly related to patient safety. And if I do work properly in my, process microbiology, I'm going to support the patients' lives. It is one of the one-day service, one-day earning, one-day service also we have for society. One-day earning also we have.
Yeah.
Earning as well as knowledge. So knowledge, earning, and service. So all together in pharmaceutical. So it is one of the good, profession. Why we should not work on the focus? It is a very simple thing. We need to focus our all senses to our work. Nothing is there. Automatically commitment will come. When we have interest, there is no time. When we don't have any interest, it's very difficult to work also.
True. I'm totally in agreement with you.
Correct.
Definitely. So, Dr. Suresh, it was immense pleasure for me hosting you and understanding your life journey. So much of knowledge, so much of, learning for me today. Thanks a lot. Of course, I'm sure, it would have impacted a lot of our viewers also. And thanks for your time. You have taken a lot of your, valuable time today. But thanks for being part of this podcast, Doctor. And for our viewers, thanks a lot for being part of this podcast. To have more understanding of the pharma, and for more latest updates and more videos and more podcasts, do follow us on LinkedIn and also download Pharma Now app today. Thank you.